So, what is it that concerns an ordinary person the most? Right. Health. And living a long life. If you have good health, because if your health is bad, living a long life isn't interesting. For example, someone has a bad heart, which is pretty common, a heart attack. The mind goes bad, the brain stops working. A stroke, a brain hemorrhage. And so on. Is it possible to grow a new heart? Instead of transplanting from a donor, waiting for months or even years, and maybe even dying without a donor heart, without donor lungs, kidneys, liver, and so on. Right now, there's a huge industry for, organ transplants. An industry, I have to say, that's frightening. In some countries, people are simply killed for their organs or ruthlessly exploited, forced to undergo surgeries for some money. A person loses a kidney, loses their eyes, and so on. That's the wrong path. But the direction that I represent, and not just me, but a fairly large community of scientists around the world, is different. So, this field is called linguistics, wave genetics. In its practical application, and its history spans more than 80 years, because our brilliant predecessor, the Russian scientist Garjajev, proposed the theory of the biological field. That was back in the 1920s of the last century. Nevertheless, he brilliantly formulated the main idea. Still, I'll give a very brief theoretical introduction, just so it's clear to you. That our chromosomes, that is, our hereditary apparatus, our genetic apparatus, has a wave equivalent. For now, let's say, we'll stop our theoretical discussion here. And I'll talk about what can already be done now, based on the use of theories and device developments that originated in the field of linguistic wave genetics, right here in Russia, in Moscow. So, for example, what have we done? One of our very first achievements was that we managed to regenerate the pancreas in animals. This was done in Canada in 2002. Then, in 2005, this was repeated by my colleagues under my supervision in Nizhny Novgorod. Regenerating the pancreas is very convenient. Instead of taking medicine or injecting insulin, you can grow, regenerate, your own pancreas. This was accomplished. Then, something else was done, though it was a single experiment. We did face certain organizational issues. We managed to regenerate teeth. That's right. And finally, one of our most recent achievements is that we actually demonstrated the transfer of a specific gene. That is, the transfer of a specific gene is possible and has already been proven experimentally. Not like we used to do when we transferred thousands of genes all at once. So, in order to regenerate the pancreas, you need to activate many genes and transfer them over a distance. And these distances are quite large. We're talking kilometers, from meters to many kilometers. Here, we developed a specific method, a technology for transferring a particular gene, and demonstrated that this gene, a specific gene called the BLBP gene, I won't spell it out, which is involved in programming stem cells. That is, we managed to transfer it remotely over several meters from donor cells to recipient cells to stem cells. Let me remind you that billions and billions of dollars are currently being allocated worldwide for stem cell research. But these efforts, I can say directly, are strategically misguided. And all that money will go to waste. So, here is a concrete demonstration. The article is already prepared for publication. And finally, the second thing, which, just like the first, just like the regeneration of the pancreas by this electromagnetic method that we developed, or rather, by the laser method, just like the transfer of the BLBP gene, all of this is also done by laser methods. Finally, our latest work is the regeneration of the cerebral cortex, the neurons of the damaged cerebral cortex. And now we are working on the regeneration of the spinal cord, on spinal cord injuries. There are a lot of patients who suffer back injuries, so-called spinal patients. There are also many people who have suffered strokes, when the brain, the cerebral cortex, is affected. As a result of hemorrhage, metabolism is disrupted, neurons are destroyed, and the person either becomes paralyzed or the neurons simply die. So, we managed to program certain stem cells, a specific line of stem cells, and introduce them into the bloodstream. And, these cells take their place in the brain, and then the regeneration of the cerebral cortex begins. Regeneration starts, and the cells begin to differentiate into the neuronal type. No one had done this before us. We were the first in the world to do this. That's why this technology has an enormous future. That is, in principle, it is possible to regenerate everything and thereby extend a person's life, prolong life, and moreover, even rejuvenate them, slow down aging. Because, theoretically, these things can be quite reasonably assumed, since there are cells in our body, there are organisms that do not age. For example, some marine organisms. And our cells, those very stem cells, they actually do not age. They can divide as many times as they want. So, the stem cell remains. It's the original one. No matter how many division cycles there are, right, no cellular death occurs, no mitochondrial death happens. These stem cells are immortal, just like, by the way, unfortunately, cancer cells. All of them have the ability to reproduce themselves for as long as they want. Thus, we have a certain material basis to extend human life indefinitely. Of course, another question arises. Is it necessary for a person to live for thousands of years? Probably, that's a rather meaningless question. Most likely, no. But it is possible to ensure an active life for a person for 200 or 300 years. If we go further, there will probably be overpopulation, and major social problems, tension, and so on will arise. But that's not a question for biologists, doctors, or geneticists. That's already a social issue. I'm just saying that it's possible. It is possible. And maybe this could be applied to certain groups of especially talented people. But again, that's not within the competence of geneticists and biologists. It's more of a social aspect. So we don't discuss that here. So, the technologies themselves are actually very simple. This is our work. The technologies look very simple on the surface, although behind this simplicity are 26 years of our research, my research. And of course, in the group, in collaboration with prominent physicists, embryologists, histologists, and even linguists, mathematical linguists. Mainly, there were physicists, because the technology we use is, of course, based on knowledge of quantum physics. So, in this way, the prospects are limitless, and, well, yes, in principle, it's possible. Many people are doing this now, and many of them speculate, many of them are at fault, but we are the most effective at recording various biologically active substances in the form of certain quantum equivalents, which can be introduced, for example, into water. Right now, this is a big problem when homeopaths dilute substances to infinite degrees, to very high degrees, when the substance is no longer present, but something still remains, and this water continues to have an effect. Or, for example, the so-called homeopathic pellets, which also, well, may have some traces of biological substances, or maybe practically nothing left at all. Maybe there's just one molecule, so to speak, among billions of other carrier substances, like sugar, for example. And yet, it still works. Why does it work? Well, this happens precisely because of what Garjajev predicted regarding chromosomes. But this prediction can be extended to all other substances as well. That is, every substance has its own quantum equivalent. If you know how to control it, if you can convert this quantum equivalent into convenient forms that you can work with, then this gives you the ability to record all medicines in a compact way as an electronic library, and the need to produce large quantities of medicines disappears. Although, in some cases, it still needs to be done, and pharmacologists do a great deal of work. I have no intention whatsoever of casting any aspersions on them. It's just that pharmacologists need to collaborate with us and with other specialists who work in this field and have the technology to record medicines in the form of these quantum equivalents. So, what is it based on? Let me touch on this a little, because it's also interesting, so that people understand that all this didn't just come out of nowhere, and that we didn't just pop up like jack-in-the-boxes and start waving around. Aha, look, we've got this kind of technology. Yes, this technology has been developed by us for 26 years, and of course, it is based primarily on quantum physics and genetics. And especially on genetics, because I want to emphasize one point. The genetic apparatus is not only responsible for passing on hereditary traits, as many people think. That our chromosomes are only there so that our children resemble their mother and father. No, that's just an important, but actually a minor part of what the genetic apparatus does. Its main function is to regulate all metabolic processes, fundamental metabolic processes that start at the level of DNA molecules with our chromosomes and end with physiological acts, physiological processes. And so, the genetic apparatus is at the foundation of everything, at the very core of everything. And here's what was done before, and what we have done. And it's very important to understand this, because right now the state of people's health may have been largely lost as a result of the fact that genetics has, in general, led us into a dead end. This is exactly the case when an insufficiently developed theory, in this case, the theory of genetic coding, has led to catastrophic results associated with GMOs, the so-called genetically modified food products, and various, so to speak, so-called synthetic bacteria that were created at the Institute of, at Craig Venter's Institute in the United States. The very same person who was the first to sequence the human genome. And about $20 billion were spent on this. So, where exactly was the mistake made by geneticists and molecular biologists that, in general, brought us to a certain threshold, beyond which lies a very promising future? This mistake, in fact, led to the catastrophic spread of genetically modified foods, which, generally speaking, cause cancer, allergies, reduced fertility, lower birth rates, and, essentially, the degradation of humans as a species. In other words, Homo sapiens is on the verge of extinction as a result of the improper use of genetic principles. And the principles of genetics were, not understood by the founders, so to speak, of molecular genetics. Let's put it that way. The founders were Nobel Prize laureates. First and foremost, that's Marshall Nierenberg and Francis Crick. In the 1960s of the last century. But it actually started even in the 1950s, when Crick, Watson and Crick, discovered the so-called double helix of DNA and thought that they had, so to speak, grabbed God by the beard. Now, they believed they understood the principles of genetics. Then Marshall Nierenberg joined this work, and in 1968 he received the Nobel Prize for the discovery of the genetic code. But actually, it was for this very model of the genetic code, not for the discovery of the genetic code itself, that would have been completely incorrect. The model of the genetic code is an approximate description of how our genetic apparatus works. The mistake was as follows. The genetic code, or more precisely the protein code, is far from being all there is to genetics. It's the program for synthesizing proteins, which are what we're mostly made of. We mainly consist of three basic components. One is water. We are made up of about 70 to 80% water. The brain, in fact, is 90% water. Then there are proteins and DNA. These are the main components, and they discovered how proteins are synthesized and encoded in chromosomes. At the same time, it turned out that the encoding of proteins in chromosomes is carried out by only about 1% of the DNA. The rest of the DNA, that is, almost 99%, was, without a second thought, called junk. That was a huge mistake. The first mistake. A strategic mistake. Well, back then it was still somewhat forgivable. Basically, they just didn't understand its role, so they called it selfish, junk DNA, and so on and so forth. Actually, this DNA has a lot of nicknames. Nierenberg made a discovery, a major discovery. He was rightfully awarded the Nobel Prize for it. But he should have either not been given the Nobel Prize or been given a double Nobel Prize. And why a double one? Here's the paradox. He made a mistake. If he had realized his mistake, he would have received 2 Nobel Prizes. But he was awarded the Nobel Prize for the discovery of genetic coding. And, without going into the details of how this coding works, I'll say that these codes are divided into 2 halves. 50%, that is, directly code for proteins, the amino acids that make up proteins. And the other 50% of these codons, which are responsible for the components of proteins, for amino acids, they code for them in a dual, ambiguous way. Here's a simple example. The word cliac, right? The word cliac. It can be understood in any number of ways. It could be a treble clef, or a key to an apartment, or it could be a spring where water comes out of the ground, which is also called the cliac. Or take the word, for example, cosa. Which cosa? A girl's braid, or a scythe for cutting grass. Or the word box, or the word block. A bow you shoot with, or an onion you eat. Or maybe it's the English word look, to see. These are the kinds of ambiguous words with, so to speak, undefined semantics, meaning, or sense. So, the entire genetic apparatus is divided into 2 halves. One half codes precisely, and the other half codes in this dual way. It could be this, or it could be that. But it's no coincidence that this was immediately called a text, though it was called a text metaphorically. So, the genes that code for proteins were called texts, genetic texts. But as a metaphor, in reality, it's not a metaphor. In fact, these are texts. And, the contradiction for the organism, when it encounters this kind of ambiguous coding, is resolved very simply. Just like how you can only understand the word cosa within the context of a whole sentence. Right? For example, I saw a girl who had a beautiful braid. Well, it's clear which kind of cosa we're talking about. Or my scythe was so well sharpened that I enjoyed mowing the grass. So here, cosa has a completely different meaning. The genetic apparatus works the same way. Using the laws of linguistics, it relies on context. What is context? It's the whole sentence. When you have the whole sentence, like with kosa, for example, then it's clear which meaning of kosa is meant. This is called contextual orientation. Simply put, a part can only be understood when you know the whole. And here is where Nierenberg made a huge mistake when he discovered that one of the codons, the so-called Regan codon, encodes two different amino acids at the same time. And the organism, or rather the genetic apparatus, has to choose one of the two amino acids. If it chooses incorrectly, and the code is the same, it's a homonym. If it picks the wrong acid, that practically means certain death for the organism. But it calmly solves this problem. How? It reads the entire phrase, the genetic phrase. It reads the whole gene as a text. And the genetic apparatus, in this case the ribosome, acts as a quantum biocomputer, a nanobiocomputer. That's a trendy word nowadays, but here it's appropriate because the ribosome, which reads the messenger RNA, is a nanobiocomputer. So it reads the entire gene, understands its meaning, and assigns the exact value to the codon. So, when Nierenberg discovered this one unique codon that codes in such an ambiguous way, they had already started working together with Francis Crick, the Nobel laureate. They observed this situation, and then openly and honestly admitted that the precise molecular nature of this phenomenon was not entirely clear to them at that moment. But even in this very phrase, there's already a misunderstanding. It's not the molecular nature that's at fault here, but rather completely different things are at play. These are the meanings of genetic texts. So, most likely, it was the mental nature, not the molecular nature of the gene, that they didn't understand. And then they simply, quietly swept this under the rug. And even Crick wrote a corresponding paper where he talked about the so-called third nucleotide in the codon, and that it could be any nucleotide, and he created a smokescreen. This was forgotten. It was forgotten for many years, for more than 40 years, because the Nobel Prize was awarded to Nierenberg in 1968. That's how it was. And since then, as you can see, so much time has passed, and yet biologists, geneticists, and medical professionals still continue to exist within this contradiction. This has led to the consequences I mentioned to you. We are now standing on the brink of a genetic catastrophe because we are thoughtlessly beginning to consume genetically modified products that are made based on an incorrect theoretical model. When artificial genes are introduced into the genetic apparatus, and these genes change their meanings according to certain acids, you end up with a bunch of other proteins that can simply kill a person. Let me just illustrate this for you and show you this table of genetic coding uncertainty. Here it is, you see. Here, the red marks indicate the so-called label nucleotides in the codons, which are exactly what give rise to this splitting of codons, splitting of genetic information, this kind of uncertainty. And these folks here, who, in fact, introduced the model of genetic coding, actually introduced an incorrect model. Yes, it's half wrong. If they had understood, if Nierenberg had realized the significance of this effect he discovered, the uncertainty in genetic coding. Here's Nierenberg, you see, that's Nierenberg, that's him. And this is Francis Crick. Then he should have been awarded not one, but 2 Nobel Prizes. And Crick should have been given a second Nobel Prize as well, because this would have been a groundbreaking discovery that would completely overturn all of genetics. But it was either overlooked or not understood, or perhaps for some other reasons. We won't find out now, because both of them, so to speak, have already passed away. But before his death, Francis Crick wrote a book called The Astonishing Hypothesis. You know, it's no coincidence that he called it a mad search. It really was mad, because in this case, they simply didn't have enough, so to speak, sense. In other words, they did it, as it were, without sense. And further on in this book, he simply admits that. In this book, there's a phrase where he says, "I don't see any obvious meaning in the genetic code." He honestly admitted it before his death. That's true. There you have it. So now, we are the ones paying for these mistakes. But linguistics, wave genetics, which I've been working on for 26 years now, makes it possible, so to speak, to lead people out of this dead end. And, well, it produces quite definite results. So, as you can see, here's this image, and here is this device. Looks pretty modest, a modest little device, right? It's a laser. A laser isn't just some little gadget, it's the result of work by Nobel laureates, Towns, Prokhorov, and Basov. Basov is one of those who worked at the Lebedev Physical Institute. I was also lucky enough to spend 10 years working in the Landberg Optics department at that institute, alongside some very prominent laser scientists. So, this is a laser which allows. What does it do? It does some really powerful things. It does some really powerful things. The thing is, our chromosomes are lasers too, in a way. They emit what's called coherent light. This light is different from ordinary light in the same way that choral singing differs from people singing out of sync, you know. So, our chromosomes emit a very weak light in a visible spectrum, that is, from ultraviolet to infrared. And at the Lebedev Physical Institute, this is our work. So, we created a laser using DNA molecules. We were the first in the world to show that. Yes, really, yes, people didn't believe us. So, scientists were skeptical because the DNA molecule seemed too complex to be a structure that emits laser light. We proved it and published our results in Quantum Electronics. By the way, this was thanks to academician Basov, who was still working at the Lebedev Institute at the time. Well, then we moved forward. So, what does this actually give us? Now we're getting closer to a practical demonstration, an experimental demonstration. What can this kind of wave-based genetic information, read by such a laser, actually do? That is, the red light from this laser, a helium neon laser, with certain quantum characteristics, strictly defined quantum characteristics related to the polarization of photons, right? So, such a sophisticated laser is truly capable of precisely reading genetic information and then converting it into a broad and expansive electromagnetic spectrum due to certain intricate processes that occur during the complex interference of counterpropagating laser beams. This is the so-called Denisuk hologram, or more accurately, running wave holography. Break time equals 0.5s/. I'll use such a daunting term, but nevertheless, it's necessary because it's at the core of our experiments. And in addition, there's another model that complements this Denisuk model, the so-called localized light model, where light is transformed into radio waves. We pick up these radio waves with a radio receiver, including genetic information recorded at the radio wave level, Well, there are also some other nuances, which we don't need to discuss right now, since this isn't a theoretical physics seminar. But here is the practical result. The result is negative. So, in what sense is it negative? It showed the power of this technology, right, which we initially encountered with negative results, and then we moved away from it. So, when we use the genetic apparatus as an information source, a damaged DNA molecule, DNA extracted, so to speak, from a particular plant, and this DNA, when isolated in its pure form, nowadays it's extracted very easily, it's a simple process, right, but in the process, it gets damaged, it breaks apart, right? Well, biochemists are fine with that, because geneticists don't understand the significance. And here is that very jump DNA I was talking about. So, when we use this kind of DNA preparation, which is essentially a damaged genetic apparatus, this is what we got. We found that these tall bars here indicate the level of mutation. That is, at a distance of 7 kilometers, we induced a mutation in the plant which, as a result, essentially dies. So, over a great distance, potentially any distance, in theory, we transmit damaged genetic information, causing mutagenesis, so-called, or more precisely, quasi-mutagenesis, because it is not associated with the breaking of DNA molecules, but rather with completely different changes at the level of this so-called excess DNA, or so-called selfish DNA. So, we forgot about the negative results. There were also some very unpleasant negative results. We realized that we needed to work very carefully with this equipment. And then came the most important results, the ones I mentioned earlier. These were obtained in the city of Toronto, where we were invited to Canada. And here we demonstrated that it is possible to regenerate the pancreas in dozens and dozens of experimental animals, and later, this could also be done in humans. I won't go into the details here. So, when you destroy the pancreas with a certain toxin, when you kill it, degrade it, diabetes develops, type 1 diabetes. And here, you see, this sugar, on this axis is the blood sugar level. The sugar level rises. Here are 4 graphs. The sugar level rises, and then the rat dies. If at a certain point in time you don't introduce wave genetic information, recorded by a laser from the pancreas of a newborn rat of the same species used in the experiment. And after that, when you irradiate these rats with these radio waves in the soft range, so to speak, the carrier wave is 650 kilohertz, and the spectrum is fractal. And then the sugar level drops. See, here at about 1 centimeter, 3 meters, 20 kilometers, 20 kilometers. At 20 kilometers, the sugar level drops. See, it dropped right away. Here too, it dropped back to the original level. We regenerated the pancreas. Generally speaking, this is, of course, a historic fact. So we published this work. It's in my 3rd monograph. Then this experiment was repeated. So, after Toronto, in Nizhny Novgorod, so the doctors I worked with. This was in 2005 to 2006. That's it. And so, here we have the regeneration of the pancreas. You see here it is. Here is the normal pancreas, the islets of Langerhans. We inject aloxin. It degrades those very islets and the beta cells that synthesize insulin, which is necessary for sugar absorption. And then we regenerate and give back, so to speak, the original picture with normally functioning islets of Langerhans. Now then, so moreover, we induced, this is also a completely unique experiment, what is called wave immunity in animals. This is absolutely unprecedented because no one, just like with gene transmission at a distance, no one has ever induced wave immunity before. But we managed to do it. If you inject a poison, for example, aloxin, but first irradiate the rat's pancreas for a long period, then this deadly poison, which normally kills rats, stops working. And these rats live on just fine. They lived for 40 days and kept on living. Now let me show you what else we had. Well, for example, you can induce resistance in people, recovery, in a large group of people. There are dozens of people involved. The difference between these 2 graphs shows that their functions were restored. Their vital functions were brought back. That's it. And this is an image that shows some very interesting phenomena related to a new type of DNA memory. This is the so-called memory at the level of the Fermi Pauli return phenomenon. I won't go into detail about this, but I'll just say that this phenomenon itself also underlies genetics. It's at the core of how the genetic apparatus works. And when it's necessary to restore an organ or tissue or even a cell, this memory kicks in, the so-called return memory. For example, a crab's claw regenerates. So what does a crab do when it loses a claw, or a lizard when it loses its tail? Right, they regenerate. To do this, they request. The organism requests the old memory of the lost limb, or say, the lost tail. Or, like with our liver, if part of the liver is removed, it regenerates. So, you need to make a request to the genetic memory, right, and retrieve the necessary information for the regeneration of organs and tissues. So, this is what we call return memory. It is actually realized even at the molecular level in the form of this Fermi-Powell return phenomenon. Moving on. So, people might ask, how are molecules read then? You say that reading takes place, that genes are being read. Yes, they are being read. There are certain wave processes that carry out the reading. You could say that reading is carried out by what's called electronic excitation, certain types of waves that occur in DNA molecules and RNA molecules. That's it. And here, I'm showing you how this laser device, yes, produces these very spectra. Here they are shown, these spectra, which carry not only genetic information, but more broadly, genetic metabolic information. Much more broadly, because the laser beam scans everything in the cell and in the tissue, including DNA molecules. So, these are the kinds of spectra we get. Here they are. Here is a case where we regenerated teeth in a woman. She had absolutely no teeth. Here is her one loose tooth, the last one. Otherwise, her jaw was completely empty, an empty upper jaw, and the lower jaw without teeth, except for this one. But when we introduced information taken from the blood of her 9-year-old grandson, who was in the process of losing his baby teeth, this information, while we were also treating her for diabetes quite successfully with this kind of wave exposure, at the same time, we managed to regenerate her teeth. And since she had no idea this was happening and was wearing dentures, the new teeth pressed against the dentures, you see, they started growing sideways. This caused her inflammation, swelling, and so on. We were quite worried about it. You could say there was a lot of concern on our part. But later, everything became clear. But this is another thing which is also connected to a particular kind of manifestation of genetic information. Our chromosomes are a kind of time machine. They are the operators of biological time. Because our chromosomes, so to speak, live in the present, look into the past, and plan for the future. That is, they operate in 3 time frames simultaneously in order to build a future organism. And here, under certain influences, like on pea seeds. Normally, if you sow pea seeds, after 3 days they sprout like this, you see? But if you affect them in a certain way, with this kind of wave-based information, their rate of aging slows down. So these are 4 days old, and these are a month old. You see, after a month, they've only grown this much. So we've managed to capture this moment related to slowing down aging. And now we're using this on people. We have a technology that allows us to slow down human aging. Well, this is, so to speak, a different phenomenon, a special one, the so-called phantom memory of DNA. That would take us too far off topic, so I won't go into it. I can only say that, for example, if you remove DNA from the device, a DNA phantom remains, and the device can detect it. This suggests that we don't just disappear or vanish. Some kind of memory of us remains in the so-called excited vacuum structures. Well, There is a certain theory about this, but we won't discuss it either. So, these are examples. Here is one type of DNA phantom. And here are phantoms of another kind on a plant leaf. When you cut off a leaf, and the shape of the leaf is an inherited trait, a genetically inherited trait, then, under certain conditions, when you irradiate the leaf, a so-called gas discharge visualization occurs, the Kirlian effect, and you can see the phantom of the DNA, oh, the phantom of the leaf. Here it is, you see, enlarged. Here it is, right here. So, we are actually seeing something that isn't there. This is exactly what holographic memory is, because with a hologram, every fragment of the hologram remembers the whole. You can cut a hologram in half, into pieces, into tiny bits. Each piece will reproduce the entire image. Thus, the demonstration of the purely holographic memory of the genetic apparatus, this holographic memory, is one of the cornerstones of wave genetics and has practical applications. Well, these are another kind of DNA phantoms, so we can do it differently. Here is a DNA sample, and if you excite it in a certain way with red and infrared light, a phantom like this shoots out of it, following a trajectory like a machine gun burst, a kind of track. And in just a second, it spins out this trajectory and then flies off somewhere. So, this is the future. This is the future of genetics. We're only just beginning to work on this. We've brought in top theorists for this. Concepts like dark matter, dark energy, and so on are involved here. We won't go into that, just noting that this isn't a seminar on quantum physics or quantum genetics. Well, here's another one of those phantoms. If you excite it differently, then this is what the phantom looks like. If you turn off the light, if you turn everything off and the lab is dark, the phantom, so to speak, continues to exist. You can't see it with your eyes. Only photographic film can detect it. This kind of phenomenon is very interesting. Well, this is yet another form, so to speak, of calculation, mathematical computation, processing of digital memory, and a phantom appears here as well. So, these are diverse phenomena that also form the basis of quantum or wave genetics, and which will need to be studied in this century and in the centuries to come. There's plenty of work to do. That's that. Well, it's a long story to tell, so here it is. DNA replicates itself. These are the so-called quantum replicas, wave replicas of DNA. And, moreover, it scans the surrounding space, which also produces replicas. This is not an artifact. This is a scientific fact. We described all of this and published it in the United States. That's that. What else should I tell you? Yes. So, this experiment is hard to see, but I'll explain it anyway. Recently, Luc Montagnier published an article. Well, it's already been a year since he shared information that shook the entire genetics world. In fact, he did what we had already done a long time ago. Only he did it. He transmitted the information using his own device, also operating on low-frequency electromagnetic fields. That's that. He didn't use a laser, because information about a DNA molecule can be transmitted in different ways. He transmitted the information over a distance and introduced it into water. He took information from a fragment of DNA of a certain length, with a specific nucleotide sequence, and introduced it into water. And then it turned out that in this water, where there was nothing except water, if you add a reaction mixture containing DNA precursors, the enzymes that synthesize DNA get tricked and accept the structure of the water, which has become DNA-like. So, it takes this DNA-like structure of water for actual DNA and synthesizes a real DNA molecule. We repeated this experiment. Unfortunately, it's hard to see here. Here is the DNA fraction, which, just like in Luc Montagnier's experiment, and let me remind you, Luc Montagnier is a Nobel Prize laureate who received the Nobel Prize for discovering HIV, the AIDS virus. So we repeated it, only using our own equipment. Yes, indeed, it is possible to transfer DNA information to water. This is a huge, so to speak, experimental achievement. Luc Montagnier and his team don't know or understand the theory behind it. We had and still have a brilliant scientist, Nikolai Alexandrovich Belyankov, whom I personally know, who established, and this is the theoretical basis for such experiments, that water can form clusters that can organize themselves into an exact likeness of DNA, RNA, and proteins. This work dates from 1982 to 1992, and, as often happens in Russia, it has been forgotten. A truly brilliant theoretical prediction, and Luc Montagnier doesn't even reference it. He doesn't even know about this publication. It was published in crystallography and biophysics journals. So, in other words, the foundation for these experiments has been established. Theoretical. And there is experimental confirmation, both by Luc Montagnier and by us. The only difference is that Luc Montagnier transmits a tiny fragment of DNA, while we transmit a huge pool of genetic information. For example, for regenerating the pancreas or for regenerating the cerebral cortex. And you can repeat the process and regenerate everything: lungs, kidneys, liver, blood vessels, and even the brain. These are the possibilities, the colossal possibilities of linguistic molecular genetics, which is fundamentally changing all of medicine. It can, so to speak, move away from drug-based treatments and instead draw information from the body itself. And here is one of our most recent, so to speak, achievements. Here's what we did, what we accomplished. So on the right is the sick girl, right? She is 8 years old. And this is her sister, younger looking, but she is 20 years old. It's just a childhood photo of her. They are biological sisters. This girl, Elisa, she suffers from a terrible genetic disease that affects millions. And in Holland, in Ireland, actually, a third, 30% of children die from it. It's simply a national tragedy, from a disease called mucoviscidosis, or cystic fibrosis, as it's also known. The Latin name for this disease is cystic fibrosis. Cystic fibrosis. This means that the gene of one of the enzymes that is involved in the transfer of sodium ions across the membrane is damaged. And this mutation is enough, and there are hundreds of these mutations, but one is especially harmful, but they all lead to the same thing, that carbohydrate metabolism is disrupted. And in the lungs, such clots form that prevent the child from breathing, and he gradually suffocates and dies. Inevitably, 100% mortality. The only thing doctors can do is use expensive medications, so-called mucolytics, which thinness mucus. The child might live for another 5 or 6 years. Or they resort to more extreme measures, right? A surgery that costs up to $500,000. A lung transplant from a deceased donor for this sick child. The child survives and might live a little longer, not 5 years, but maybe 7 or 8 years. But eventually, they still die. So what can be done? The gene is damaged. The body can't do anything about it. We did something that is basically magic, but quantum magic. And there's even this Sergei Ivanovich Doronin, an absolutely brilliant physicist, our Russian physicist. He works at the PDI. He wrote an amazing book called Quantum Magic. All of quantum physics, it's all magical. I don't have the time, and there's no need to tell all these fantastic things here, but I'll mention just one, because it's fundamental and probably the most cited, so to speak. It's the article by Einstein, Podolsky, and Rosen, written in 1935. Its title goes something like, "Can quantum physics, describe the world in its entirety, so to speak?" In it, they postulated one thing. It's purely formalized, very much in the realm of physics and mathematics. It's very hard to find fault with it. In this regard, it's absolutely correct. So, based on, let's say, theoretical reasoning and physico-mathematical formalism, they prove that elementary particles, which sometimes exist in what's called an entangled state, for example, photons, when these elementary particles, like electrons, photons, and so on, fly apart, they are in what's called an entangled state. That is, they are essentially like a single photon that has been stretched over a vast distance. If you change some quantum characteristic of the left part, the left photon, for example, polarization, which, by the way, is key for our work. So, if the left one changes its polarization, the right one instantly responds, without any time delay, and changes its polarization in the opposite direction, but in exactly the same way, spin, so-called spin up, spin down. So, it turns out that distance doesn't matter at all in this case. These photons can be separated by an infinitely large distance. They could be at opposite ends of the universe, and yet they are somehow connected to each other. This is what frightened Einstein, because it violated the principle of causality. Because, as we know from general logic and from the general, and from the philosophical concept, first comes the cause, and then after some time, the effect. Cause and effect. But here, cause and effect are merged together. That is, the left photon changes its quantum characteristics, and the right one instantly changes its own in response, with zero delay. In a mirror-like way, of course, no matter the distance. So, this really puzzled Einstein. And he never fully understood it, even as he passed on to the next other world. But it turns out, as we suppose, yes, genetics uses this principle. It uses photons, after all. Photons transmit information from cell to cell. There are a lot of ways information is transmitted, from slow, very slow, to very fast, and even, more precisely, instantaneous. That is, our cells, and the genetic apparatus of each cell nucleus with its own set of chromosomes, is in what's called an entangled state. They are connected to each other by so-called wormholes. It's not a very nice term. I don't like it, but no one has come up with a better one. And they say entangle, entangled state. So each cell knows everything about every other cell instantly. But do you see what the problem is? Knowing everything is as good as being dead. That's why these states are quantized. That is, only part of the information is taken, and depending on what the organism needs, it uses this quantization. Have you seen the phantoms I showed you? They are all quantized. So, maybe this is a reflection, in particular, of these quantized states. This is absolutely necessary. Why, then, is quantum locality needed for the transfer, in quotes, of information between cells? Because we have cells, and their number is absolutely staggering. About 200 billion cells. And the communication between them not only has to be fast, it has to be instantaneous. Only then will the biosystem be whole. Although there are slow processes as well, in certain modes there are fast, ultra-fast, or more precisely, instantaneous processes at work. So what am I getting at here? So what do Alice and her sister have to do with this? Take cystic fibrosis, for example. It needs to be cured. It needs to be cured. There's also another concept, a grand one as well, put forward by Bohm, who said that the universe is a hologram. Everything in this world is holographic. As above, so below. Even the ancients used to say this. He pointed out, as a physicist, he said that the universe is a hologram. And every person in this hologram is, so to speak, printed out, smeared throughout. In a hologram, There isn't a specific place where objects occupy their own spots. Everything is blurred. But you can connect to this hologram if you know how to connect. And we, intuitively, purely through experimentation, discovered this thing. It seems like magic, but I repeat, it's quantum magic, as Doran wrote. So we take, and people shouldn't be surprised, because this actually works. And, in particular, here it worked brilliantly. We take a photograph of a healthy person. In this case, to heal this girl, we took a photograph of her healthy little sister. She's about 2 or 3 years old here as well. Extracted information from her, quantum information, obtained the so-called running wave hologram according to Denisyuk, multi-frequency, and so on. And apparently, the mechanisms are still unclear. This still needs to be worked out. But we connected to this universal hologram, took this information, recorded it as a spectrum, and gave it to the sick girl to listen to. These files, which we recorded in a simple MP3 format that cuts certain frequencies, compresses, and so on. I was criticized for this. It's nothing serious. These spectra are fractal, self-similar, and any fragment, just like in a hologram, reproduces the entire spectrum. After a few days, this girl fully recovered. And now I'm waiting for the genetic analysis of her chromosomes, because she was diagnosed with a mutation of a certain type, right? A certain nucleotide is affected, which means there's another abnormal one in its place, and so on. So, we're waiting for the genetic analysis. This is a unique and singular case where I'll be completely satisfied with either outcome. Let's say the mutation is gone. There are mechanisms that allow the mutation to be removed. Thanks to so-called transposition of DNA segments, it's possible to cover up the damaged section to replace it with a normal one. That's one option. If we find this, well, it would be excellent confirmation of our ideas. But let's say the mutations remain, yet the child is healthy. Then a major theoretical challenge arises for genetics to explain this. Why does the child have a mutation, and a genetic analysis, which will be done in the next few days, shows that yes, the mutation remains, but the child is healthy, even though the gene is affected. The linguistic component of genetics explains this very simply. It works on the principle of it's written as Varenka, but read as New York. Just like the word kosa. Which kosa do you mean? A girl's braid, or the side you use to mow grass, or a bow you shoot with, or the onion you eat? Do you see what I mean? This affected gene is a homonym, and it can be understood one way or another depending on the context of the surrounding, so to speak, DNA structures. And these DNA structures are variable. So, in genetics, Barbara McClintock received the Nobel Prize, I think in 1968, for the discovery of mobile genes. That is, genes jump, they jump around. Why do they jump? What controls them, these jumps? Where do they jump to? This is still not understood. But the Nobel Prize was awarded, although, when she discovered these jumping genes, realized that they jump, she did this on corn. This was somewhere at the end of the 40s, beginning of the 50s. People thought she was a crazy old lady. But in 1968, she was awarded the Nobel Prize. Even now, people still don't understand what the point of gene jumping is, but the significance is enormous. Linguistics and wave genetics say that these gene jumps achieve two results. The first one. So, when genes shift, when DNA jumps, and the sequences of nucleotides change, the hologram changes. This change of holograms is absolutely essential for the organism. Yes, both during embryonic development and simply throughout the life of an adult, it is absolutely necessary. This is achieved through this, well, transposition of genes. That's one. Second, the contextual landscape of the genes changes. And once the contextual landscape of the genes has changed, as probably happened with Alice too, and soon we'll know for sure, the meaning of the gene, the effect of the gene mutation, flips to the opposite. There can be a mutation, but the organism doesn't even notice it. You see how it works? It says Marinka, but it's red as New York. That's what a linguistic wave of genes is. That's its practical application. All genetic disorders can be cured using the methods of linguistic wave genetics. And all the unfortunate people who suffered radiation damage, hours after the Chernobyl nuclear power plant explosion, now Fukushima in Japan, so many affected people. All of them can be cured if we properly use the principles of linguistic wave genetics. So, you see, this is direct proof that the theory is not just a good theory, but a practical one. And finally, as a little treat, I'll give you something interesting. So, in 2002, we were working in Canada, in Toronto. In a place called Chilbolton, this kind of image appeared. You know, nowadays there are so-called crop circles, but in this case, it's not circles, it's what's called the response to Carl Sagan. So look here, here's the picture enlarged, here it is, you see? And this is its diagram. This is what was on the wheat field. This is the classic DNA, which, among other things, was sent by Carl Sagan to the M9 constellation. And so, it came back 27 years later. The picture returned with a very strange DNA molecule. See this here? DNA like this can't function. And what does that mean? This is a pictogram of DNA radiation. So these guys are saying, folks, you've driven genetics. Genetics has driven you into a dead end. You're handling DNA molecules the wrong way. You don't understand that DNA is both an emitter and an antenna for genetic information. And they're showing us. It's like we're at an exam. If we manage to understand it, great. If not, we're just trapping ourselves in a dead end. Right now, humanity is at just such a stage, but linguistics and wave genetics allow us to get out of this hole and live long and happily to be healthy. That's roughly what I wanted to say about our work.